We're going to get started here for the afternoon session. We've got let's see, we've got Eric.
Yeah.
Yes, and Moshe here. Great, and everybody's ready to go. All right, are we live on? Ready to go live? Great. All right, I'm going to turn it over to you all. Thank you all.
Awesome. Well, thank you for coming back after lunch, and for this our second session, which is called Innovative Non-Immune Therapy-Related Therapeutics and Monitoring. So we purposely separated out, again, things that are not immunotherapy related, not because we don't love immunotherapy, but we want to sort of think about tumor cell autonomous processes and how to actually really understand how those can be targeted therapeutically, as well as looking at ways that we can monitor what's happening in the microenvironment as well as in the tumor. So this is sort of the raison d'etre for these awardees, DoD awardees. And it's my pleasure to have Moshe Ornstein co-moderating this. And he's, as you all know, assistant professor at Cleveland Clinic and head of the kidney cancer program there.
So thanks, Eric, and thanks to the organizers for inviting me to moderate. It's really exciting to be able to see what might be the next clinical breakthroughs in RCC in its pure scientific state, which is where we'll see most of the information today. So our first speaker is Dr. Mei Yee Koh, who is the founder of Kuda Therapeutics, but also on faculty at the University of Utah. She's done extensive research in hypoxia and HIF inhibition, and she'll be discussing the investigation of ferroptosis as a novel therapeutic paradigm for kidney cancer.
Thank you. So good afternoon, everyone. I'm really excited to be here. My first in-person meeting, and just seeing everybody and talking to live moving people is really, really exciting. So today I'm going to talk about ferroptosis, a novel form of programmed cell death that we pursued at Kuda Therapeutics, a small biotech company that I founded, that we've kind of stumbled serendipitously on a new strategy for targeting kidney cancer. So those of you are all familiar with clear cell renal cell carcinoma. It has its characteristic clear cell appearance as opposed to the uninvolved, which is more less clear cell. And the clear cell appearance is due to a metabolic alteration in the cancer cells that are VHL and HIF dependent. Essentially, to summarize a broad area of research, essentially VHL loss induces the accumulation of lipids in the cytoplasm of these cells due to the inhibition of mitochondrial fatty acid oxidation. So with VHL loss, which is the initiating component of clear cell renal cell carcinoma, you have this lipid accumulation. And when you reintroduce VHL, you lose that. That's the bar chart on the right, where you see when you introduce VHL, you lose oil redo, you lose lipids. So why do we care about these lipids so much? For us anyway, we know that lipid accumulation sensitizes cells to a new mechanism of cell death known as ferroptosis. So this is a form of programmed cell death that's distinct from apoptosis, so it doesn't require caspases, and it shows necrosis-like morphological changes. The rate-limiting substrate for ferroptosis are the presence of polyunsaturated lipids. So 3 hallmarks of ferroptosis, just to introduce the group, are polyunsaturated fatty lipids, fatty acids within phospholipids, redox active iron, and defective lipid peroxide repair. So you need iron, you need lipids that can be oxidized, and you need a defective lipid peroxide repair system, resulting in cell death via ferroptosis, which, as you can see in the picture on the slide, are characterized by little dysfunctional mitochondria and accumulation of reactive oxygen species and iron. So a new form of cell death. This is the only pathway picture I'm going to show, and it's way more complicated than that. But for the reasons of this talk, I'm going to summarize it as the pathways involving antioxidant response. So I guess I have to point at it, the glutathione pathway. I'm going to point through Eric. So the glutathione synthesis pathway right in the middle, and on the left side, the phospholipid synthesis pathways. And so you have the major pathways that we'll talk about are GPX4, which inhibits ferroptosis, and iron, which promotes ferroptosis. So you have all these other pathways that drive into ferroptosis, but the points that we care about for today's talk is really that the presence of lipids sensitizes cells to oxidative death through ferroptosis, iron catalyzes this effect, and GPX4 opposes this effect. And why is this exciting? So it's been shown that drug-tolerant persistent cells, so cells that are resistant to apoptosis, are still vulnerable, sometimes more sensitive to ferroptosis, mediated by GPX4 inhibition. So GPX4, like I told you, protects cells against ferroptosis. Multiple groups have also shown that CCR, actually all clear cell tumors, but clear cell renal cell carcinoma particularly, are hypersensitive to ferroptosis and GPX4 knockdown. And this is driven by HIF2, which we know is a validated target for clear cell renal cell carcinoma. Finally, it's also been shown that CD8 T cells induce ferroptosis during PD-L1 blockade. So the CD8 T cells actually inhibit the glutathione antioxidant species, promoting ferroptosis as well. And the combination of ferroptosis inducers with PD-L1 blockade causes a synergistic effect in promoting tumor cell death. So what we see as the unmet need, however, is the fact that, well, we all know that we've made incredible strides in the treatment of kidney cancer. The combination therapies, immune checkpoint inhibitors have really pushed the boundaries of patient response and survival rates. But as we discussed before this morning, there's far to go, we can do better. And ferroptosis, we think, is a new strategy that can potentiate the effects of immune checkpoint inhibition. However, the current ferroptosis inducers, Erastin, RSL3, and a number of others, although they induce ferroptosis very well in vitro, they don't do very well in vivo. They're not good starting points for drug development. They're not stable. They don't have good bioavailability. So these are not good drug candidates. And so this is the work that we've initiated, funded by the DoD grants and also by the NH, NCI, and SBIR phase 1 grants, is that we've discovered a lead molecule that can, in our search for inhibitors of HIF2 alpha, we found that these molecules also decrease levels of GPX4, the molecule that protects against ferroptosis and induces ferroptosis. So the first slide, picture shows a Western blot showing our compound decreasing HIF2 alpha and GPX4. The next one shows comparison of KD025 with PT2385, which is a predecessor of the current Merck compound. And we know that this compound inhibits HIF2 alpha very well. But we also know that these compounds, the PT compound does not promote cell death in vitro. So we see no effects of the PT compound on cell death, and our compounds kill cells. So at first we thought this was rather, non-specific, you kill cells. What does that really mean? And we delved deeper, and we found that these cells were dying in a mechanism that was preventable by either an antioxidant like NAC, you see in that chart, or by the iron chelator DFO. But it was not preventable by the apoptosis, the caspase inhibitor, ZVAD, FMK. So we have an iron and oxygen-dependent mechanism of cell death. And if you talk to ferroptosis researchers, we also had to show that we promote lipid peroxidation. And so that's what this bar chart shows here, a shift in the fluorescence of FITC of the BODP 581-591-C11 dye, which validates that cells are undergoing lipid peroxidation and iron-dependent cell death. So this is the only data slide I'm going to share today. In the interest of time, I'm just going to say that we found the molecular target of our drug. It's known as iron sulfur cluster assembly 2, ISCA2. It's a bit of a mouthful, but essentially it's a relatively unknown protein involved in iron metabolism. And so we think that we've come across a new strategy to both target HIF2 alpha and induce ferroptosis. So our hypothesis, at least for the DoD grants, is that ISCA2 inhibition It's a novel and promising strategy to both block HIF-2 alpha, a known tumor driver in clear cell renal cell carcinoma, and to promote ferroptosis. So inhibit tumor growth and promote tumor cell death. Our aims are really to improve on KD025. It works quite well, but we want it to work better. So the DoD has funded us to perform medicinal chemistry SAR approaches to improve the drug-like characteristics of 025. And with this new compound, we plan to investigate the in vivo efficacy of this strategy, either alone or in combination with currently available PD-1 inhibitors. So our goal really is to assess the feasibility and utility of this therapeutic approach for kidney cancer. So to take home what I've just said, the induction of ferroptosis is a promising therapeutic strategy which targets the intrinsic metabolic vulnerability of clear cell renal cell carcinoma. That's HIF-2 alpha driven lipid accumulation. Ferroptosis is recognized as a novel and exciting anti-cancer strategy, but current ferroptosis inducers are suboptimal and are not good for drug development. And what we propose is a new strategy from a high throughput screen, starting with 025 as a novel orally available small molecule to serve as a starting point for further lead optimization efforts. Our big goal, our impact, we hope in the future, is that this would generate a new lead molecule to test this first-in-class therapeutic strategy that can actually selectively target the lipid accumulation that is a metabolic hallmark of kidney cancer and induce cell death in drug-tolerant apoptosis-resistant cells and synergize with currently available TKIs and immune checkpoint inhibitors. So with that, just acknowledge our team. We're a small team. Carol Santos, a research associate, done fantastic work. And we have our chemist, Xiao Hui, and another lab assistant, John Brodsky. Acknowledge Eric and a number of other people in our SAB as we build this small company. And the video you see is a prostate cancer cell undergoing apoptosis. So with that, I'll take questions. Thanks so much.
Hey, Mei, just very quick question. So how does the KD025 target HIF-2 alpha for degradation or decrease HIF-2 alpha levels?
So that's the whole new research grant that I have actually. It's a new molecular pathway that we've discovered. So essentially, long story short, is that we inhibit mitochondrial function and that impacts the synthesis of HIF-2 and actually HIF-1 later on. And it's not through degradation. We're not quite sure which way. It might be through IRE-dependent translation of HIF-2, but we still need to validate that. That's my question.
Right behind you. Actually, mine is a related question, and I think you just answered it, but there's a known relationship between HIF-2 alpha, iron homeostasis, and so it's quite likely that you're affecting the translation of the mRNA that encodes it. Do you guys have any data along those lines?
Yeah, and we do affect IRE-dependent translation, but the part that I didn't show is that we have some effects on HIF-1 as well. Also not degradation, it happens later on. So in the world of research, it's always really fascinating in the world of trying to develop a drug. We don't quite know yet, but that's at least one mechanism why we block HIF-2.
Dr. Tenier?
Yeah, great talk, Mei. Do you have any data to show that this strategy, ferroptosis induction, is not just limited to tumors like clear cell with lipid accumulation, but it is independent of lipids at all? And this may be a mechanism actually that can be seen in many tumor types and not clear cell RCC. Because the next talk in our session will be by Dr. Lisa Hensk about ferroptosis as a possible valid target in patients with chromophobe RCC. So my second question, have you tested your compound in UK 267 cell line, which is a chromophobe cell line?
Well, the short answer to the second question is no, we haven't tested it in that cell line, but I did get a chance to talk to Lisa at lunch as well. So what we're going for with clear cell is the low-hanging fruits. And that was because Stuart Schreiber's group did a screen, and they screened a whole variety of cancer cell types. And they found that cancer cells with the clear cell morphology are most sensitive to ferroptosis inducers. By that I mean RSL3. And among the clear cell types, clear cell renal cell carcinoma is the most sensitive. This does not mean that any other cancer cells are less or not susceptible to ferroptosis. It was a product of a high throughput screen in cell lines. And so this was a starting point. I'd like to add to that, we did reintroduce VHL in all these cell lines that we tested with KD025, and VHL does promote resistance to the ferroptosis caused by our compounds. So there is a VHL dependence of the sensitivity, at least in clear cell, but we haven't tested this in chromophore or any other cell.
So Eric, if you're online, do you have any data about this in primary tumors and not just cell lines?
No, but we'd love to try if we can get some primary tumors.
I think if we have time.
One quick question. In the Nature Com paper we had with Stu Schreiber, we also looked at clear cell ovarian cancer, ovarian clear cell. Is this another strategy that you're exploring too?
Not at the time, but this is something that will be on our horizon. We want to have proof of concept in a model that is validated and that we have expertise in, and then we'll build on to other models. Thank you.
Great. Thank you so much.
Thanks.
Phil. In the interest of time, we'll move to the next speaker, Dr. Celeste Simon, who is from the University of Pennsylvania, where she's a professor and serves as the scientific director of the Abramson Family Cancer Research Institute. Her extensive research topics include cancer cell metabolism, and today she'll talk about exploiting cholesterol metabolism to treat primary and metastatic RCC.
Yes, I walked over starting about an hour and a half ago. So good to see everybody. These are my disclosures, and maybe I'll join your group as the previous slide indicated. So we appreciate that most solid tumors, if not all solid tumors, exhibit extensive genetic diversity and intratumoral heterogeneity at the genetic, epigenetic, and of course, metabolic levels. And clear cell renal cell carcinoma is no exception to that rule. And nevertheless, we've asked a question that we think we're making progress on is do disparate oncogenic changes converge on more common metabolic adaptations, and might they be actionable? And so we took the logical integrative approach. We banked our own tumors many years ago. We performed copy number variation studies. We combined this with transcriptomics, proteomics, and now we've embellished that with the extensive publicly available transcriptomic and proteomic data. And putting this all together, we've been looking for very consistent findings. And ones I won't talk about today, which we're very interested in, have to do with the gluconeogenic pathway and the urea cycle enzymes, which I could talk about if anybody's interested over a beer, perhaps. But as you heard, and everybody here knows, these are extremely lipid-laden tumors. And they've been diagnosed for decades based on an incredible capacity to accumulate cholesterol, cholesterol esters, and triglycerides. But why is that? And so we looked at our integrative approach with metabolomics, lipidomics, and transcriptomics, and we're actually quite surprised to see that the intracellular endogenous biosynthetic machinery was completely suppressed. We can see this at the level of the enzymes, but also as indicated here, and I'll try not to point through Eric, one of the key steps in cholesterol biosynthesis, squalene. This metabolite is significantly underrepresented in the tumors compared to the adjacent healthy kidney. Now, something that's quite interesting, and I'm learning more about this, but working with colleagues in the United Kingdom in a process known as Mendelian randomization, and I'm just learning about this, so bear with me, but the notion is that you can look at extensive GWAS studies and figure out causative relationships between phenotypes and whether the phenotype is upstream or downstream of observable component of the serum. And this was looking at a number of patients with high BMI and noting that out of 135 evaluable metabolites, HDL cholesterol accumulation is elevated in the serum, and this actually is predisposing to individuals with clear cell cancer, not the other way. And I'd be happy to talk about that a little bit more. So why is it that the tumors are accumulating so much cholesterol and cholesterol esters, which is causing them to suppress the endogenous machinery? You know, as you heard, they're very lipid droplet laden, and we've shown previously that this makes them strictly dependent on developing lipid droplets. They're dependent on perilipin proteins. And if you have high levels of HDL cholesterol, you're going to be that much more likely to develop this disease. So why have we seen this, and why is this the phenotype very consistently of these tumors? And we've learned that the tumors very consistently overexpress the surface protein that's a key mediator of HDL cholesterol particle import. It's called SCARB1, or also scavenger receptor family member B1. We've shown that this is almost invariably overexpressed in the tumor material compared to the healthy kidney. And we wondered if it was needed for continued tumor growth. At this point, we've done this in simple subcutaneous flank tumors. So, you know, we understand the caveats of that, but very simply, if we eliminate the expression of this protein, the tumors, we let the tumors get to a certain size and they basically shrink away, as indicated here. So we think that this gives us a new therapeutic opportunity. The reason being that SCARB1 happens to be a co-receptor for hepatitis C virus. People have developed antivirals, and these are, didn't mean to do that. Can I go back? Oh, good, I can. Okay, so these are two different examples, and the on the right-hand side, the ITX5061 compound has actually been taken deep into clinical trials as an antiviral, but then those studies stopped. We don't believe that that's because of any problems with the patient. Unfortunately, we think the company went out of business, but this compound is available, and we've been able to try it in both our in vitro and, more importantly, in our in vivo models. So this is something that we think could be translated to the clinic relatively quickly. And our current studies, which we've been funded by the DoD for, is we're now implementing this first in PDX approaches at Penn. We're working with our surgical colleagues to implant PDXs through the prescribed protocols that probably many of you are aware of and perform yourself, and try the drug. More to the point, we'd like to put this into humanized PDXs. So these are patients whose immune systems or their hematopoietic system has now taken on, or I should say these are mice. These are our patients, mice are our patients. These are mice that have human tumors, but also human immune systems, because we suspect that this consistent cholesterol accumulation may actually be contributing to decreased efficacy of the immunotherapeutic approaches. So it's now clear that, wow, I still have 7 whole minutes to talk. I don't think it advanced. So just shut me up when the time is right. But it's very clear now that if T cells get exposed to cholesterol, it can promote exhaustion. So the idea would be if we could decrease cholesterol accumulation, especially in the tumor interstitial fluid, could this perhaps not only help with the controlled growth of the tumor itself, but potentially augment immunotherapeutic approaches. So just to summarize, as I said, this is a very consistent metabolic adaptation that everybody here is already aware of. This propensity to accumulate cholesterol esters. We believe that the reason why they're doing this is because they overexpress the SCARB1 protein. We also believe that this is a considerable metabolic vulnerability for these tumors or these tumor cells, because by accumulating all this cholesterol, they downregulate the endogenous biosynthetic machinery. They become dependent on lipid droplet formation. And moreover, as indicated right here, they have to deal with the buildup of oxysterols. And so now they engage another pathway, which is bile acid synthesis. So that's led us to yet another very consistent protein that's very consistently overexpressed, called HSD3B7. The enzyme's real name would take me 7 minutes to pronounce, so I'll just call it by its acronym. But this is a key step in bile acid synthesis. And I'll just mention that clear cell patients tend to have a considerable amount of bile acid in their urine, almost as if they've suffered liver injury. could be a biomarker, but we believe that this is another dependency, and by similar approaches, we've shown that the tumor cells are equally dependent on SCARB1 as they are HSD3B7. And the final question is, so why do they do this to themselves? Why do they so consistently overexpress this HDL cholesterol importer? And one suggestion we can make that I don't believe for a minute is that this allows them to access the AKT pathway. I actually think SCARB1 is doing something else very important. We just don't know what it is. So ongoing CRISPR screens are trying to discover what that might be. And this is how our lab parties look in the age of COVID. And so people like Nico Schooley, Fuming Lee, and he's not showing up, Rindert Missian contribute to the studies I presented today. So I hope I didn't go over my time. I think I might be okay. Be happy to take any questions.
Questions?
Thank you.
Very nice talk. Maybe you touched the point and I missed it, but do we know what is the effect of the introduction of statins for, hypercholesterolemia in kidney cancer incidence?
Right. So from my read of the literature, it's been very equivocal. And what I can tell you for a kidney cancer cell itself, you could imagine that the statins would have no effect. They've completely downregulated that pathway. The statins do have effects on the growth of normal related kidney epithelial cells, especially those that haven't been grown in the presence of cholesterol. But in the epidemiologic data, I think the findings to my read are very equivocal, and that's beginning to make sense to me because the tumor cells themselves shouldn't be dependent on this or shouldn't be affected by the statins per se. But if the statins affect circulating HDL cholesterol levels, they would. And so I would say it would depend on when the disease is discovered, with whether the statins would really be efficacious. I still hold out considerable hope for this other idea we have because it would be irrespective of that observation, but that's how we see it. Lisa.
Are you hearing me? So why do the cells not utilize the lipid and cholesterol they're storing and become dependent on SCARB1?
So I think it's actually maybe the other way around. I actually think SCARB1 is doing something else that we don't understand yet. And that's why, we could explain it that they express SCARB1, it gives them a growth advantage because this seems to be a way to contribute to the PI3 kinase AKT mTOR pathway. And of course, we know mTOR dysregulation is a hallmark of the disease. But I suspect there's something else going on. And so because they're so dependent on SCARB1, this causes them to import all of that HDL cholesterol. They're absolutely cholesterol oxytropes. We can show this in vitro. If we try to grow the cells without cholesterol, they can't grow at all. But they do imbibe it. And then because of their intrinsic mitochondrial deficiencies, and we know that the beta oxidation pathway is extremely underexpressed, maybe for redox reasons, they don't use it. So they store it, they detoxify through bile acid synthetic approaches, and they downregulate the endogenous pathway. So it looks like a big fat metabolic vulnerability just waiting for us to take aim and shoot it, is what I would say. So that's what we're trying to do.
Two questions online here. First one says, great talk, agree with that. Is this new inhibitor toxic to normal cells? In other words, what's the therapeutic window?
So at least from the clinical trials, trying to deploy this as an antiviral seems to be pretty well tolerated, at least based on the data that we can access. It hasn't been tried yet in renal cancer patients, so the therapeutic window in that setting isn't clear to us yet. We're trying to do a couple of preclinical studies for due diligence and then working with our colleagues at Penn to really try to get it into the clinic. GPX4 knockout is lethal in mice. I agree.
Let's get
a quick question. Do you have information on whether there's VHL selectivity for this SCARB1 receptor or
is
the
there any
inhibitor?
relationship to SCARB1 HIF2? So that's
Yes.
another reason why when we saw it was overexpressed, we were very intrigued. because GWAS studies from a decade ago from people like Steve Chanick and others have shown that there's a SNP in the gene that encodes SCARB1, and that SNP just so happens to create a novel HIF2 alpha binding site, at least in the patients that have that polymorphism. However, we don't believe the gene is expressed by the or regulated by the VHL HIF system because we can add back VHL or take away HIF2, nothing happens.
Maybe we'll take one more from the screen from Steve Zeiger. While we are waiting for more research on SCARB1, should kidney cancer patients consider a statin?
So I don't know how to answer that question, to be honest with you, because like I said, you could imagine some benefit, but I fear that once the disease has been diagnosed, the cow's out of the barn, so to speak. You know, it could lower HDL cholesterol levels to some extent, but I don't know that it would do it enough to really help the disease progression itself. I think this could be more beneficial perhaps to people with the germline VHL mutation. As I said, this has been looked at in epidemiologic studies, and I think the data is pretty equivocal. So I guess I would say I wouldn't necessarily recommend it with high hopes.
Well, thank you very much. In the interest of time, we're going to move on. Our next speaker is Dr. Scott Haake, Assistant Professor in the Division of Hematology and Medical Oncology at Vanderbilt, where his research focuses on immune monitoring and biomarkers. His talk is titled Monitoring Disease Burden and Biology Using Tumor Cell-Free DNA in Metastatic Kidney Cancer. Scott.
Thanks so much for that introduction. And thank you so much for the opportunity, the organizers, to be here and share with you some of our DoD funded research. So when we conceived of this project, our goal was to develop a liquid biopsy or a cell-free DNA tool that we could use to monitor disease progression as it either grows or shrinks. And so the clinical model that we decided to utilize was monitoring metastatic kidney cancer patients while they receive immunotherapy, with the thought that as their disease grows, we should be able to see increases in our molecular biomarker, and as their disease responds, hopefully, which is the real hope, that we see a parallel decrease in our liquid biopsy or molecular biomarker. And so just to reiterate, our hypothesis was that tumor cell-free DNA, which was the molecule which we chose as a promising candidate to monitor this clinical response from patient plasma, serves as a robust and dynamic marker of immunotherapy response as well as progression in kidney cancer patients. And so time points, I won't belabor the details in terms of how we chose these time points, but ultimately, as part of the trial, we're collecting patient samples at 0, 3 months, 6 months, and then at either 24 months or time of progression. And this is specifically a patient blood. And so this to me is the most exciting part of the talk, where we go through the different technologies that we're pursuing with the goal of being able to differentiate the tumor DNA that's released into the patient circulating plasma and use molecular features of clear cell renal cell carcinoma to distinguish that cell-free DNA that we find in patient plasma from the normal cell-free DNA, which is found in all people all the time. And so obviously, if you're in the field of circulating tumor DNA or tumor cell-free DNA, a first good stop on your journey is looking at disease-focused somatic variants. And there's been very nice work by people in this room, Dr. Powell and colleagues, where they looked at a panel of somatic variants and sequenced the cell-free DNA in kidney cancer patients. And this was relatively successful. They were able to find molecular aberrations in the vast majority of their patients. This is an important proof of concept that you are able to retrieve cell-free DNA from patients and do molecular analyses which will detect kidney cancer. And so we've developed a similar panel. We've developed 10 and 20 gene panels. The one careful thing that we have to consider when we're wanting to monitor disease burden over time is intratumor heterogeneity and clonal evolution of kidney cancer. You can imagine if you start at time point A with tumor A, some tumors shrink, but then new clones grow out. Unless your somatic variants are shared among all clones within this patient, if, say, for example, your panel only detects somatic variants in tumor A, which I've pictured here, you may be showing a response in the patient, whereas in fact they have a mixed response or progressive disease. So for this reason, it's very important that we focus on truncal genomic aberrations, which are shared by all clones. This is going to be the most easy to understand biomarker. Obviously, one of the molecular hallmarks of clear cell renal cell carcinoma is loss of function of the VHL gene, but it's a very hard gene to sequence. As shown here, it was mutated in approximately a third of clear cell renal cell carcinoma patients. And so when we started thinking about developing this biomarker, we wanted to think about what are truncal molecular aberrations of clear cell renal cell carcinoma that all clones would share, and thus would be most easy to use as you follow a patient over time. And so fortunate for us, there is another molecular hallmark of clear cell renal cell carcinoma, that being 3P deletion, which is thought to be the first step in the molecular tumorigenesis of clear cell renal cell carcinoma. So could we design panels where we're specifically looking at 3P deletion in tumor cell-free DNA and monitor the amount of cell-free DNA that has this deletion over time, and use this as a biomarker to monitor patients? And so when we started this journey, we're like, well, let's sequence all of 3P, go really deep, and normalize this to a stable portion of the genome. And for the sake of time, I won't show this data, but it turns out it's very hard with that strategy to normalize, to understand what diploid is. And so that was not a successful strategy. And so what we pivoted to was sequencing not only all of chromosome 3, but as the title of the slide suggests, doing whole genome sequencing. This helps us to establish diploid within the tumor cell-free DNA. And this is a strategy that we're currently pursuing. So this was an experiment where we were piloting some of these techniques, and we took A498 human kidney cancer cell lines. This is a 3P deleted cell line, and we extracted genomic DNA, fractionated it so that it approximates cell-free DNA, and then when we performed dilutions into a healthy donor cell-free DNA to determine the level of sensitivity of this assay. And as you can see from the slide, using our next-gen sequencing platforms, we were able to detect cell-free DNA as low as 5%. And if you plot the estimated tumor fraction versus the known dilutional percentage of cell-free DNA, it was a rather linear relationship. So we were encouraged by this. And this is just a handful of patient samples. So the one on top is a healthy donor where it's diploid throughout, which was very encouraging. These are two of our first patients that we've sequenced, where we were able to detect aneuploidy. It's important to note that this assay doesn't just detect 3P deletion, but it's really a global aneuploidy assay. We also pursued a complementary technology, which I won't have time to give much detail on, but suffice it to say that the technique is called droplet digital PCR. We designed probes within the region of 3P, which is commonly deleted, as well as reference probes outside of 3P. And in theory, it worked quite well. However, the sensitivity was somewhat limited in that when we did our dilutional experiments, at a 1 to 10 dilution, we weren't able to observe the 3P deleted DNA anymore. Also, the input for this assay tends to be much higher than our NGS-based platforms, and so we've pivoted away from this technology. One of the real advantages or perks of working within the DoD-funded Kidney Cancer Research Consortium was being able to collaborate with investigators across the country. And so we're very excited to be able to collaborate with investigators from Dana-Farber, who have pioneered this cell-free methylated DNA IP approach, where the nice thing about this is that there's a purification step where you're able to enrich for DNA which is likely from tumor. And so the basics of this approach is there's an antibody-based immunoprecipitation step where you pull out the methylated DNA, and then you're able to distinguish based on methylation patterns what's from normal tumor, or excuse me, normal cells and what's from kidney cancer DNA. And so and this is a robust platform that's been well published and well received and is very successful at discriminating between normal and tumor DNA. And so we're working with our collaborators to develop quantitative techniques where we can do this over time and measure dynamic changes in the cell-free methylated DNA. So right now, again, as I mentioned, we're working within the Kidney Cancer Research Consortium. We've currently accrued patients from Vanderbilt, the Nashville VA, as well as MD Anderson, and I think today are shipping some samples out, some sample collection kits to UT Southwestern. So we're excited about that, and we hope to bring on some other sites soon. So far, we've collected samples on about 36 patients, 27 of which have multiple time points. So we've gone ahead and extracted this DNA. We're getting good amounts of analyte back. We only need about 20 nanograms to do somatic variants, low-pass whole genome sequencing, as well as the cell-free methylated DIP. And so we've got plenty of material to do those 3 assays, plus extra material should other opportunities arise. So again, just to reiterate, these are the time points, and I've mentioned the platforms which we're pursuing. And so I am out of time, but just to summarizing, we're leveraging truncal tumor-specific genomic alterations to quantify tumor cell-free DNA over time in kidney cancer patients. These are metastatic patients receiving immunotherapy. I want to thank the patients and the families who are participating in the trial. Great mentors, Eric, as well as Kim Rathmel and Brian Reaney and Ben Ho Park back at Vanderbilt, as well as our collaborators at Vanderbilt led by Katie Beckerman, and also our collaborators at Dana-Farber, Dr. Friedman, Dr. Schwery, and team who are going to be doing the cell-free methylated DIP. So thank you again for the opportunity.
Yeah.
Thank you. Very nice talk. Quick question. So according to the TracerX study, it seems that, for example, the loss of 3P, since you were talking about the loss of 3P, happens decades before the emergence of the clinical disease. So do you think with this technology, you would have enough sensitivity to catch, something that is not, that you cannot visualize by MRI or CT, but you can visualize with circulating DNA or even DNA in urine, for example?
Yeah, sure. So that's certainly our hope. The proof will be in the pudding per se. But the only reason that you wouldn't be able to detect dynamic changes is if there's so many cells with 3P deletion, which are shedding DNA, which overrides the tumor cells, which are doing the same thing. And I would predict that not be the case. I would predict that the rapidly proliferating cancer cells are going to contribute most of the 3P deleted DNA. And if that were the case, then maybe the cell-free methylated DIP assay is the better assay. We feel fortunate to be able to test multiple assays.
We have an online question here. Why look at features shared by all clones? Bill is asking this question. Can one use cfDNA to track clonal evolution?
So absolutely. I mean, and we are tracking somatic variants over time. And so one thing we're curious about is, are there different somatic variants at baseline versus at time of progression, and in responders versus non-responders? I think all these will get at clonal evolution over time. But the important thing to remember is, at least for the value that we see in this assay, is being able to monitor all disease burden over time. And so the only way to do that is to target molecular aberrations, which are shared by all clones. And so it just depends. So you design the assay to meet your needs. If you're wanting to monitor global disease burden over time, there's one strategy. If you want to look at clonal evolution, there's other strategies, and they're not always overlapping.
Scott, can I ask you a question? What about, obviously, the idea of adjuvant therapy? And what about doing these assays preoperatively and then again, postoperatively, kind of taking a page out of the bladder cancer textbook?
Oh, man, that bladder cancer data is beautiful, isn't it? That is just so inspiring. And so certainly we think that would be a clinical utility. My primary focus is on the technology and finding the best assay that works in kidney cancer. And then the way I see it is, if you find a useful tool, just like Apple has developed the iOS system, and anybody can build apps for it. We have a lot of really smart physicians and researchers in this room. Get the tool working and we'll find clinical applications for it. That's really low-hanging fruit. And I think that paper by Powell and colleagues is inspirational. That's what we should be doing.
We'll take one more.
Yeah, unfortunately, the bad news here, I hate to be the Debbie Downer, is that renal cell does not shed really much DNA. So I don't think the success that Dr. Powell's and colleague had with their Nature paper is even gonna come close in renal cell. And fair enough, the same test used in the Nature paper, the bespoke assay, investigator actually at Fox Chase with Philip Bosch tried to do that, and sensitivity was only 40%, because really it doesn't shed much DNA RC. So hopefully with CF methylated DNA-seq will be able to do better. The one thing also that maybe, you know, a differentiator here is that doesn't use bisulfite sequencing, so you can do mutation. And the second thing, we have data in urine that looks less good, but we're trying to optimize it. So I think, unfortunately, I don't think the data in bladder gonna apply in renal, but we're hopeful.
I think it's important to draw inspiration, and the proof will be in the pudding. So a couple caveats there. There's adjuvant versus metastatic, and so it's hard to know whether tumor shedding changes with tumor evolution. And we're averaging about 100 nanograms per patient right now. And we made a very simple change. Instead of collecting 1 tube, we collected 4, right? So not a difficult problem there. And so we're averaging about 100 nanograms per patient. The cell-free methylated DIP platform takes about 10 nanograms. Our low-pass whole genome sequencing and somatic variants, we're currently running at 10 nanograms. So at least for the assays that we envision, they're possible. Will they be sensitive? Will they be dynamic? We don't know. But
your approach of combining multiple platform is the one that gonna probably work, because, you know, one size fit all with the bespoke assay is probably gonna have low sensitivity. It's still gonna detect, but I don't think we're gonna have the same effect as in the Nature paper. But that's great work.
Well, we look forward to seeing the results. Thanks, Dr. Our next speaker is Dr. Abhishek Chakraborty, who's a assistant staff at the Cleveland Clinic Learner Research Institute. And after spending about a decade in Dr. Kalin's lab, he brought his talents to Cleveland, where his research focuses on hypoxia, epigenetics, and cancer metabolism. Abhishek.
Might as well ask everybody their age and like how long they've impolite questions to postdocs. Okay, how long you've been in the lab? All right. My lab now, finally my own lab. We continue to develop some of the things that I originally started doing in Bill's lab, and I have no disclosures, not important enough as yet. But what I do a lot is think about ways. Very generally to try and target renal cancer cells, typically clear cell renal cancers in the lab. And a very simplistic model that we think about a lot is you have a tumorigenic state, which is the VHL deficient state, and a non-tumorigenic state, which is the VHL proficient state. And can we now look for epigenetic changes, metabolic changes, or any other variations of interest that you could perturb in an unbiased manner and ask what's the biological significance? And so typically you could do this with like a negative selection screen, like a necessity screen using CRISPR-Cas9 tools, or a sufficiency screen where you put back genes that are typically down-regulated in a VHL proficient cell and ask which ones are sufficient to confer tumorigenic ability. We've been doing this for a lot of time, right? But the one thing that we have begun doing a lot in my lab is moving to more in vivo screens, which allow us to read out critical candidates a lot better than growing cells in culture. So one sufficiency screen that we did in vivo some time back was using a gene list that we had mined using epigenetic changes in VHL proficient and VHL deficient states. So that's the H3K27 acetylation mark, which we had found upregulated in VHL deficient cells. And we had overlapped this with transcriptomic changes and asked what genes, so typically you do this, you'll get thousands of genes actually scoring And then we said, we want to look at genes that are going to be subsequently usable in the clinic. So we listed the top 100-ish genes which could encode druggable products, and we asked in a sufficiency screen, you put back these genes in VHL proficient cells and which ones would now score as oncogenes. Okay, so long story short, each row is a tumor over here. When you see the cells are in blue, that means those genes disappeared in the tumor. They are not acting as good oncogenes. The ones that are in red are actually behaving as oncogenes. And so now you take these individual candidates, put them back and ask, are these true hits from your screen? And a couple of them did indeed score as true hits. One of the ones that we have consistently seen in multiple different cell lines is a transporter called SLC1A1. It transports aspartate and glutamate into ccRCC cells. So this was really interesting because aspartate and glutamate are both non-essential amino acids, and everybody wonders why the hell should this transporter act as an oncogene? And really the critical reason for that is normal cells and tumor cells really have very different nutritional demands. And so what's non-essential for a normal cell is really not non-essential for a tumor cell. These cells have to constantly generate enough biosynthetic product for them to divide efficiently. And as a result of this, in a number of different oncogenic studies, you'll find that these transporters score as critical dependencies, including some that are in the same SLC1A1 family. So we've gone and we have begun testing functionally what this gene actually does in kidney cancer. Jess Minara is the postdoc who's done most of this work. One of the first questions she asked was, is this gene even necessary for the survival of the ccRCC cells? And very simply, she used CRISPR tools. She inactivated SLC1A1 in ccRCC cells, and she asked, does that have a deleterious impact on survival? And it actually did. Every time you can eliminate SLC1A1 efficiently, you see a fitness defect in those cells. But anybody who's been in this business for as long as I've been will tell you that this is easy. You can kill cells in a petri dish by throwing bleach on them. Doesn't mean anything. Oftentimes, it's better to see if you can rescue the cell, and that actually teaches you something really cool about the biology. Over here, we did that. We put back either a functional form of SLC1A1 into cells or a non-functional form, which is called the RW mutant over here. And what you can see is that the wild type rescues, but the functionally dead mutant does not. So it is a canonical role of SLC1A1 that's driving its oncogenic potential. So then what do we do with this? So why is it doing that? It takes up all of these common non-essential amino acids in. Can any of these amino acids be the critical players? And so we said, okay, let's take a cell that does not have SLC1A1 and make these amino acids enter the cell artificially. So you used esterified versions of these amino acids, and we asked, could we rescue now the fitness defect? And it was pretty amazing. We didn't expect this to work as well as it did. You could try any number of amino acids, non-specific controls, as well as the critical ones that are known to be substrates for SLC1A1. And aspartate was the only one that we were able to rescue consistently the fitness defects associated with SLC1A1. And so we are now thinking about what exactly is happening. Why is aspartate so critical? And so we have an ongoing collaboration now with Dr. Vanderheiden's lab to try and figure this out using some flux tracing type experiments. So what do we do with this knowledge? Is it going to be usable? So on one hand, we are trying to work to make some pharmacological inhibitors that can target SLC1A1. But there's already evidence in the clinic that could help us test whether this could be usable in ccRCC. So for example, many people in this room would probably know, although the clinical trials were a little bit disappointing for everybody, but nevertheless, glutaminase was thought to be a potent dependency in ccRCC because it can convert glutamine into glutamate. There are inhibitors available. CB839 is a really famous one. And we said that theoretically, the function of SLC1A1, its ability to take up glutamate and aspartate from the extracellular space, would counteract the dependency on glutaminase. And we tested this out functionally, and you can do this in both different directions. So you can take cell lines that do not express SLC1A1, you put back SLC1A1 into these cells, and now you can make the cells resistant to the glutaminase inhibitor. That's the panel on your left. You can do the opposite experiment. You can take cells that express SLC1A1 highly, and you inactivate SLC1A1, you make the cells sensitive to the glutaminase inhibitor. So now from a clinical standpoint, you have to think, why are these patients not responding? Are there biomarkers or in fact functional drivers like SLC1A1 that could be telling us that, well, there are other metabolic ways by which the cell can get around this problem. And therefore, this gives you the rationale for combining SLC1A1 inhibitors down the line with the glutaminase inhibitor and chemosensitizing the cells in that way. So this is just a very quick last minute sort of idea. And we have been thinking about this quite a lot in the field in general. So just the way we are what we eat, the tumor cells that we grow in the lab are also what they eat, really. And the soup in which we grow them really does not look anything like the condition under which they're physiologically surviving. And there are people who are actually trying various different approaches to address this problem. So we now have synthetic formulations based on what the metabolite composition of plasma looks like. And so there's one composition from E.L. Gottlieb's group, which is, they call it the Plasmax composition. David Sabatini's lab published a separate one. They call it HPLM, or human plasma-like media. And we have to ask whether these media changes are going to affect the metabolite state of a ccRCC and then affect the dependencies that we score when we test these conditions. And largely, if you take the bird's eye view, we don't see a dramatic difference when we tested this out. We're quite surprised by this. But actually, if you poke and look for specific details, you can actually find that there are indeed metabolic differences depending on how you grow the cells. And in fact, some of them are probably physiologically relevant, actually. So Celeste is sitting over here, and she'll tell you immediately that, oh, wow, urea cycle intermediates, many of them. And they have a story that they've published about how these are critical down-regulated metabolites in some, from a clinical standpoint. Although again, recently I've looked at some studies in bioRxiv right now, where that seems to also be context dependent, and depending on stage of the tumor, the urea cycle is either is up or down regulated. But the reason why I'm bringing this up is because as a community, we need to discuss about what is the relevant condition under which we should be doing these experiments. And now to add an additional tier of complexity, Matt Vanderheiden's lab had gone one step further and said, well, plasma might not be the most physiological state to do these experiments in. Every tissue in the body really has a completely different interstitial metabolite composition. And so maybe the kidney TIF might down the line, which I think the Vanderheiden lab and I think Kim Rathmill's lab are together working on, and that down the line, that might be useful for us to start thinking about even screens that we are doing. So I'll stop there and I'll take any questions. These are all the people who have done the work. Jasminara has primarily spearheaded the work in my lab. Sam McBrayer and Ben Olenchuk were my metabolism gurus in Bill Kalen's lab. And then we have a fantastic team of collaborators in Cleveland who are helping out with this project. So I'll take any questions.
Great talk, Abhishek. Just wondering, CBS39 is not effective in ccRCC clinic trials. Do you think that's actually primarily due to this LC1A1 expression of regulation?
Yes. So basically, it's critical to define what exactly was done, right? So the trial was advanced RCC and no difference, no significant change in overall survival of metastatic RCC patients. So already these patients had undergone pre-treatment and several rounds, and that's a caveat that we should recognize. But yes, coming to your specific point, so we are working with Calithera already to see if we could now go back and look at their tumor samples, maybe responders and non-responders, and see if SLC1A1 status could be a biomarker to differentiate the cohorts of patients. That's exactly what we are working with them right now. Yes. Yeah, yes.
Your screens, are you doing them under ambient air?
Okay, so that's a great question. So we had a conversation about this at one of our offline meetings. Typically, the in vivo screens, of course, we're not worried about this. We're just injecting into either orthotopically or subcutaneously. We have tried to do the screens both with just ccRCC lines in a hypoxia chamber, but also with like organoids of RCC in a hypoxia chamber. We find that the organoids cannot tolerate hypoxia at all. Like even 15% oxygen, they cannot survive in that condition. But maybe that's okay because the organoids are already in like this extracellular matrix capsule, if you will. And so they're probably experiencing hypoxia physiologically even under that culture condition. But we have not, we are thinking about doing this. We just haven't had the bandwidth to do it as yet, to do exactly what you're saying, to test out the dependencies at 5%, 2% oxygen. And in fact, do that take it one step further and do it in HIF deficient and HIF proficient lines. Because if these dependencies are HIF independent, then we might want to score what are the hypoxia driven dependencies, but regardless of HIF status. Yeah.
Any other questions?
Yes.
Great talk. Did you test the combination of the drug CD839, telaglenastat plus your inhibitor preclinically as a screen to see if the combination is better than single agent of these?
Yeah. So great question. So that's kind of the ration. That's the direction that we are going in, and that's kind of why we did the genetic experiment first. The chemical inhibitor is a really bad molecule, and so that's why with Sean Stauffer's group, who's a medicinal chemist at Cleveland Clinic, we are trying to develop a more specific molecule that targets SLC1A1. But that really is going to be our eventual goal. We want to combine the two molecules and ask. Currently, what we are doing, and that's kind of also part of our MTA with Calithera, is we want to go in vivo with CB839 and ask whether SLC1A1 function is going to impact outcomes in vivo. So we are already doing the in vivo experiment with the genetic inhibition of SLC1A1. The pharmacological drug-drug interaction, we want to do that. We just don't have a good molecule to confidently do it as yet.
Great. Thank you very much.
Sure.
So this ends our session. So
Too
hopefully
early?
now you're convinced that tumor cell autonomous treatments in renal cell carcinoma are part of our future, as well as tumor cell autonomous measurements. So thank you very much.
Great. Thank you.